Morning everyone, and welcome to the A Light of Touch Soilborne Disease webinar series. Session 1 today with Dr Len Tesoriero from New South Wales. Today's talk is on why soilborne disease is so challenging. This is the first in the webinar series of 4. My name's Jeff Smith. I'm the technical lead for A Light of Touch, looking after the agroecology projects. Just a little bit of housekeeping for the webinar today. If you could submit questions, do that at any time, but please use the Q&A function in the Microsoft Teams toolbar. If you've got a question or you'd like to like a question, please do that because it will promote the question up the list so that we can prioritise the questions that we ask to the presenter. If the Q&A is not available, you can email questions to the news@alightoftouch.co.nz email. So today, just to introduce Dr Len Tesoriero. We found Len through an AusVeg seminar actually, when he was presenting on soilborne diseases and cucurbits. This is a particularly relevant topic for our own buttercup squash project. And that project team have gone on to engage one-on-one with Len and certainly found his expertise to be of much value to that project. In this session, Len's going to set the scene for us, explaining what soilborne pathogens are, how they work, and why they're so challenging in our cropping systems. I'll just a short bio to introduce Len to the webinar. After a career as a plant pathologist with the New South Wales Department of Primary Industries, Len retired in 2018 and established the consulting firm CropDoc Consulting Pty Limited. He maintains a role with the New South Wales DPIRD as a visiting scientist, contributes to disease surveillance and mentoring younger plant pathologists. Over the past years, Len has conducted research on disease of almonds, pistachios, olives, and vegetables. He has previously worked extensively on integrated disease management strategies for protected cropping vegetables and cut flowers. So I'll hand over to you, Len. Thank you very much.
Thank you, Jeff, and thank you everybody for your attendance. Today's presentation, if we go to the next slide, I'll try and kick us off with a bit of an overview of the presentation that I'll be delivering. Thanks, Gina. So thanks for the introduction. Defining what a soilborne pathogen is, and I guess putting it in perspective, the fact that there's this strong interface with the soil, with the root, the rhizosphere and what we call the rhizobiome. I'll be highlighting a bit about some of their lifestyles and how they're important, and why they're difficult to manage, largely defining some of their survival structures, which allows them to be maintained within the soil profile. And I'll give plenty of examples from everything from viruses, bacteria, fungi, chytrids, oomycetes, things called plasmodiophyrids, which are the things that are similar to oomycetes, but they're a distinct group, and nematodes. Next slide, please. So these plant pathogens, they're microbes, and they're part of the normal ecology and environment within the soil structure. They can directly infect seeds and roots, and a few of them actually are able to be foliar pathogens. So they'll produce spores that can actually go up and infect leaves, flowers, stems, and fruit. One of the key things I think, and when we define what these soilborne plant pathogens are, is this distinction between soil inhabitants, which survive for sometimes many years, decades even, and others called soil transients, which have a shorter term survival in soil. One thing I have become aware of, though, that some of these soil transients, there is a continuum, and you can find, and as we have found, some of these things that aren't supposed to be there for very long at all can survive over 12 months. So I'm not sure what that actually, like I said, it's a continuum rather than a strict cutoff anywhere in particular. Next slide, thanks. So why are they difficult to manage? Well, one of the reasons, and traditionally we've had access to some agrochemicals that have been used. We've used fumigation strategies to actually reduce their populations in soils prior to planting. And some of those aren't available any longer. I remember in potatoes we used to use products like quintazine and things like that, but you can't get those chemicals anymore, at least not in Australia. So a lot of them have been deregistered, and they're pretty nasty chemicals. Going back, there are even heavy metal type chemicals available for use against some of these pathogens. But one of the key things is that they're difficult to monitor and identify before they've done significant damage. And I guess that's led to people looking at ways of testing soil prior to actually planting to determine whether or not the populations are there and whether they've reached some sort of threshold that can give rise to disease. And that's tempered by the fact that the numbers vary significantly in space across the field and with soil depth. For example, I've seen some evidence and some research where they've shown that the clubroot pathogen in brassicas can go down a couple of metres. You can find it a couple of metres in depth. So, any treatment that you might be applying in the top 30 centimetres isn't going to be completely effective. And these numbers vary a lot in time, and I'll go on a little bit in a few other slides to show some examples of this potential for rapid inocular multiplication. And that's another reason why when you go out and soil sample and you can do PCR tests and things that can give you an estimate of the numbers of the pathogen in the soil, it's a pretty tricky proposition to get that accurately and to, I guess, be able to predict what might happen over the next weeks and months when the crops are put in. Another factor is that many of these pathogens have a wide host range, including weeds, and some can survive and multiply as saprophytes, and some even become endophytes on plants. And a good example of that was where I was looking at Fusarium wilt in cucumbers, and quite often a lot of our growers would reuse the same media and they would grow tomatoes as the following crop. And I was able to isolate the cucumber Fusarium in tomato plants in the following crop up to about 20 centimetres up the stem, which means they weren't causing any disease, but this particular pathogen was able to survive in this tomato crop. So that's another example of it's a little bit more complicated than we often think. Next slide, please. You'll have to forgive my drawing here. It's pretty crude, but I didn't want to pick one off the web. Look, the other big thing I was talking about is what we define as the rhizosphere and the related rhizobiome, the complex group of organisms. And I know the next presentation will be about nematodes. I've said worms, but nematodes are certainly a big part of this rhizosphere population and the biome. And we know that roots are leaking various chemicals, nutrients that provide this population of organisms sustenance, and sometimes even the sloughing cells that you can see at the bottom there of the root cap provide an environment in which these things survive. And as you can see, it's a complex web of organisms, all interrelated. Next slide, please. Which brings me back to this diagram that I put together a few years ago, which I guess puts the plant back in the centre and plant health in the centre. And some of the key things that, obviously influence plant health I've listed here. And for those, I guess, who are importantly looking at say tree crops that are grafted onto rootstocks, and that's something in the work I've been doing in almonds. I've been able to, I've been enlightened, I have to say, in terms of some of the quality of rootstocks that can have resistance to things like root knot nematodes and the like. So they're pretty important in that space. But most important, I think, it comes back to the soil physics, chemistry and biology, that whole thing about the rhizobiome and how it interacts with the soil structure and the quality, what you might define as soil health. So that's one of the key areas. I'll also, during the presentation, talk a little bit about how plant nutrients are important in terms of disease management, and a couple of strong examples of where some soil-borne pathogens are influenced by the plant nutrition. Next slide, please. Let's start off with some viruses, and this may not be as relevant to some of you, but certainly it's become a big issue in Australia as with some of these tabamoviruses have been found in the last few years. And I think it's back in 2012 I found this Tombus virus as well. And interestingly, and I mean the biology of these things are amazing, that these tabamoviruses are very stable in the environment and in soils, and so is this Tombus virus. So really important pathogens because of their ability to both survive, and they're very contagious, so they're highly transmissible. There's some others that, again, are not so relevant in Australia. I'm not sure about New Zealand, but some of these nematode-transmitted viruses are important around the world. And another one that has found its way to Tasmania in potatoes is the potato mop-top virus. And I think that is in New Zealand, and that's spread by Spongiospora, which is one of those plasmodiophorids I was talking about. So not a true fungus, but an interesting group. And then there's a few others. This Mirafiori virus in lettuce, not a huge problem, but it can be. I think there's a lot of genetic tolerance or intermediate resistance to this sort of virus in lettuce nowadays. But it's spread by a soil-borne chytrid called Opidium. And similar to that one I mentioned in the previous slide, melanocrotic spot virus is also spread by a chytrid, Opidium bornivanus. So both of those things generally occur together. And then there's a whole mass of phages, these bacteriophages that actually attack bacteria, and they can actually be our friends in that they can help reduce the population of certain plant pathogenic bacteria. And they're really quite numerous. They and nematodes are some of the most numerous organisms, if we call viruses organisms, in that rhizosphere and part of that rhizobiome. Next slide, please. So those tabamoviruses I was mentioning, they're very stable. They're all pretty small. When I started, this is the methodology we used mostly, which was an electron microscope to visualize them. And given some of the problems that have been occurring with some of the molecular techniques in Australia in detecting the tomato brown rugose fruit virus, and internationally, I might say. I listened to a talk from the UK about it. It would be useful to employ some of these older techniques to actually validate some of the test results, I think. They're seed-borne and they're highly contagious. They most notably can be spread and survive as aerosols. So that's something that's really problematic, in particular where you've got a greenhouse environment and you're needing to clean up at the end of a crop where you know that the infection has occurred. And there's a whole family of these viruses, and excuse me, thankfully nowadays the seed companies are finding resistance, sometimes only intermediate resistance, but hopefully over time more robust resistance, as they have found to some of the older viruses like tobacco mosaic and tomato mosaic. Next slide, please. There's some images of some of those viruses I was mentioning, the lettuce big vein, the pepper mild model, which is one of the tabamos, and the melon necrotic spot virus. So quite a severe symptom, at least in those melons. Next slide. So let's look at some of the bacteria, and many of them are considered to be soil transient, so their survival tends not to can sort of be measured in months rather than years. But we've been looking at Xanthomonas campestris, which is the cause of black rot disease in brassicas, and we've found that survival probably greater than 12 months. We stopped the previous trial at 12 months, so there's certainly potential for these things to survive for maybe a couple of years, and so that obviously is important when you're choosing rotation crops, and if you're looking at sort of a planning for future cropping, so those things can come back, and most of the survival seems to be with decaying organic matter, so that's another factor that is important. Maybe breaking down organic matter as rapidly as possible might be a way of reducing that survival time. Bacterial wilt, I'm not sure if it's present in New Zealand, but we certainly have it in Australia, and there are strains of it where I've worked in Asia that are quite severe and have devastating effect on a lot of different crops. So there's many different strains, and nowadays I think they've even split the species. Soft rot bacteria, another group. Again, we used to call them all Lawinias, but nowadays they've been split up phylogenetically into different genera and species, and sometimes subspecies. So there's a whole host of those. They also become problematic in transit and post-harvest. They can be a major issue as well. Crown gall and the agrobacterium group are obviously important in crops like grapes and some of the fruit trees and ornamentals. We'll go to the next slide where I'll sort of talk a little bit more about these, and just again showing you that electron microscope image there on the left shows that these things are pretty small. They're 1 to 2 microns in length, so that's 1 to 2 thousandths of a millimetre. We see them when we diagnose these diseases. Quite often they ooze out, and you can see that sort of massive, basically a massive bacteria, so billions of them, and there's some colonies growing on agar. One of the important considerations about bacteria is that they live in populations, and not all of the genes are present in any one individual, so they can actually move genes around within a population, and they are far more sophisticated than we ever thought they were, in that they actually do communicate with one another. They have a system called quorum sensing. It's all chemical based, and they can actually understand, or when I say understand, it's probably bit strong, but they can actually sense when the population is building up, particularly inside plants, so they can actually take they'll switch on genes that'll produce enzymes and things that can be important in the pathogenesis response. Next slide, please. And again, this is an image probably from the American Phytopath Society. It's an older one, so it's again not quite all that accurate in terms of some of the names, but I mentioned Agrobacterium, crown gall, and some of these other things like hairy root, which actually down on the bottom right there, it's got rhizobium in quite a separate group. The Agrobacterium and rhizobiums are actually much more closely related than it shows in this diagram. Clavibacter, which is an important one, both in potato ring rot disease, and I've been working over the years on tomato canker. So that's an important one that globally for tomatoes, particularly in greenhouse cropping. And then the soft rot bacteria here, I've mentioned there's Pectobacterium, Dickia, as well as Erwinia. So there's a whole range of genera now that are known to be causing various soft rot and things like black rot disease in potatoes and the like. So there's quite a range of those, and they've sort of used to be lumped together under Erwinia, but they're now known to be different. And there's a few that would be of biosecurity importance. I think Dickia solani is present in some of the cooler areas in the northern hemisphere. I don't think it's found its way to the Antipodes at this stage. The Pseudomonads, again, most of those are more important as foliar pathogens, so I won't talk much about them. They're not really, if anything, just soil transients. I mentioned already black rot, Xanthomonas campestris, and there's a few like the onion bulb rot pathogen, which I'm not sure how widely spread that is in our countries, but again, there are a few that are more known to be soil-borne. And then the streptomycetes, so the common scab fungus in bacterium in potatoes, and a few others that are also beneficials. So there's a mixed group within the streptomyces group. Next slide, please. So there's canker in tomatoes, as I said, one that I've spent a lot of time working on in the protected cropping industry in Australia, and one that every so often flares up because it can also be seed-borne, and if a bad batch of seed turns up, it spreads like wildfire. And there's probably been several cases where globally there have been outbreaks of this particular disease. It's actually not a great survivor in media, so it is more mechanically transmitted than through media and through irrigation systems, but it is known to do that. Next slide. Here's the soft rot bacteria. So as I mentioned, there's a great number of those. Again, when I was working in Asia, it was in sort of hot, wet climates, really important. One of the interesting things about these bacteria is there's a lot of passive spread with various insects. So that's another thing, that it both attracts insects as the tissues got this rot. And I was reading an interesting paper where there was actually a gene that was important for the survival of some of these bacteria in the gut of a fly in the northern hemisphere. And where this gene occurred, this particular fly was able to transmit the bacteria. So it's really a clear association between, I guess, the actual bacterium, its survival in the gut of this fly, and the vectoring of the bacterial disease. So it's one that, again, I think would be worth investigating further down in our environments where these bacteria can be problematic. Next slide, please. So now we come to the fungal and the fungal-like pathogens, and again, this is a huge group, obviously, and genetically quite diverse as well. So it's hard to kind of pay sort of justice to them. The example of the one there was botrytis, and we don't think of botrytis as being a soil-borne pathogen, and largely it is a foliar pathogen, but it does produce these sclerotes. So on the left-hand side, the lower left-hand side of that life cycle there, you can see the sclerotia that they're the survival structures, and these are survival structures produced by a lot of different fungal pathogens, and that is the key for their survival over long periods of time. But also, it enables us to think about microbial biocontrols that might be able to parasitize them. So that is important. So we don't think of botrytis as being a soil-borne pathogen, but it can be, and you can see the bulb rot in onions there at the bottom. But mostly we think of it more in terms of a blossom blight and a fruit and foliar disease. Next slide. We'll start at the beginning, I suppose, with soil-borne diseases. So damping off is one of the key ones, and there's some images of some of the ones that I've come across. So there's sort of damping off of seedlings, but then, that causes those plants, if they survive the cotyledon stage, sometimes to not be very productive. And I've been working in more recent times with baby leaf crops like spinach, and that's the one with the blue background there. And I am finding sometimes a range of different pathogens associated with that root rot disease. So it becomes more like a root rot succession, or in some cases a disease complex. So there's things like the oomycetes, Pythium and Phytophthora, Rhizoctonia, which is a basidiomycete, a sort of mushroom type relative, and Fusarium, which is more in the kind of group of the Ascomycete group, and Aphanomyces, which is related to both Pythium and Phytophthora, so it's in one of the oomycetes. Next slide. One of the other things when I was looking at lettuce grown in hydroponics was that a lot of the Pythiums, and you'll probably be aware or have heard that Pythium's a noted root nibbler. So it basically chews away or infects some of the smaller roots, and quite often, unless you actually see, as you can on the left-hand side, a healthy plant, you wouldn't really know that there was too much wrong with those plants on the right. But you can see the colour of the roots is slightly brown, and there's far fewer roots there. At this stage, those plants are not wilting and dying. So again, you can get just a reduced plant vigour result from it. Quite often, Pythium root rot diseases are referred to as Browning root rot, and that's probably evident in that image, particularly the slight browning of the roots. So that's what I would call a minor pathogen in the Pythium sphere. There's over 100 different species, so there's many different types. Next slide. So one of the key things when we talk about things like Pythium and Phytophthora, and to some extent that Aphanomyces that I mentioned, is and I mentioned it earlier when I talked about rapid multiplication. These guys in a wet environment can produce motile spores called zoospores, and they're a weird looking thing that you can see those what we call flagella, which are the thing that push them through the water. That one on the left-hand side and the blown up image on the right shows these little twirly hairs on that flagellum that push it through the water. It's very energy requiring, and it probably only evolved once, that particular type of structure and so that's one of the reasons why, when they were looking at the phylogenetics of this group, the Oomycetes, they've sort of placed them into a separate group called Strumenophiles. They're quite a unique group. But yeah, the main things I wanted to show was that those zoospores can multiply the population of the Pythium or the Phytophthora or the Aphanomyces by a factor of 100 in a few minutes. So if you've got the right environment, that is free moisture in the soil or waterlogged conditions, these guys will multiply up very quickly, and that's when problems occur with the roots. So you might see that in the next slide, please. And there you can see those zoospores attacking either the root tip or on the right-hand side where there's been some damage to the root itself. The zoospores are collecting there in that image. And the other thing that these guys do is that they've got survival structures, so these things called oospores. And that's important for two reasons. One is that those oospores can survive in the soil environment for long periods of time. And the second is that these guys are sexual spores, so they actually give rise to genetic variation. And so it's another factor for these Oomycetes that enables them to adapt to different environments. There's some genetics that can be put into play. And even in the case of some of the Phytophthora, there's cases that are known now where they can even hybridize different species, can actually reproduce. The overall implication of that may not be great, though. Next slide. Fusarium is, I guess, one of the ones I've done a lot of work on. That was my PhD topic in cucumbers, and very serious disease in protected cropping. But I'm currently working in melons and looking at some of the former speciales, the subspecies of Fusarium oxysporum that cause wilt diseases in either watermelons or rock melons in Australia. A really interesting group in that they have these subspecies that have, I guess, what we would say are what they call former speciales, which means that they actually have a limited host range, and so the cucumber Fusarium that causes wilt doesn't cause a wilt on watermelons, but interestingly, when I tested it, it did cause wilt on rock melons. So Cucumis sativa and Cucumis melanus, I guess they're more closely related, and that's one reason why the Fusarium was able to attack rock melons as well as cucumbers. But one of the curious things about them is that they have all the genetic arsenal to get inside, and as I said earlier, they can cause disease in cucumbers, this one can, but it can also get inside the vascular tissue of crops like tomatoes, so without causing any disease. And what they found is that there are a set of genes that are actually on little chromosomes called supernumerary chromosomes, called 6 genes, and 6 means secreted in xylem. So they're only secreted in the xylem, and if they have the right combination of these genes and the products of them, then they will actually cause the vascular wilt disease. So that separates out some of the subspecies of this Fusarium oxysporum. The other important thing is that they produce little things called chlamydospores, which are the survival structures in soil. And again, they can survive for several years. Next slide. Hybrid, I guess if you're a brassica grower, you'd be concerned with this one. So this is a Plasmodiophora. That's the thing I called a Plasmodiophorid, one of these things that are more related to slime molds than to the true fungi. and they also produce zoospores and as part of their life cycle. And something I forgot to mention with Fusarium, and it's relevant here for clubroot as well, is that that interaction with nutrition. So for Fusarium, the form of nitrogen is really important in terms of the disease severity. So for Fusarium, if you use the ammonium form of nitrogen, so if you're side dressing with urea and the like, or using mixed fertilizers with a high level of ammonium, that will actually increase the Fusarium wilt symptoms and the actual it actually favors the disease. So one prevention measure is perhaps to use a nitrate form of nitrogen rather than ammonium forms of nitrogen for Fusarium. For clubroot, it's been shown that nutrition and pH can be quite important in reducing the symptoms. And in fact, pH, bringing the pH up to about 7.2, and calcium and boron nutrition can be quite important in reducing the severity of clubroot disease. I did some work overseas in Asia on cabbages, and we found a combination of that as well as some fungicide application was quite useful in reducing the disease. Next slide, please. There's a whole range of plant pathogens that are soil-borne that produce sclerotia. So I mentioned botrytis before, the white rot fungus that occurs on onions, and then there's several other. It's kind of weird. The Sclerotium cepivorum is in the Ascomycete group, but there's another one that occurs in warmer climates. So I'm not sure how important it is in New Zealand, but it's in the United States. It's called southern wilt, which again suggests that it's in the warmer areas of the United States where it's important. We get it. I've seen Sclerotium rolfsii. It's got another name as well nowadays, but it's actually quite unrelated to Sclerotium cepivorum, and we get it in Queensland in various crops like capsicums, and it's got a wide host range, tomatoes, capsicums, all sorts of things. It gets into carrots and you name it. It can be a very wide pathogen, whereas Sclerotium cepivorum is quite specific to the onion family. But quite a serious one, particularly where there's limited rotation of crops. It's an important one. But those little black sclerotia, I think you can just see them on the right-hand side, affecting those poor root systems there around the base of the plant. Quite often you'll see them developing. Next slide. The other one that's sort of similar, I guess, in the sclerotial forming is sclerotinia, and it has, I guess, a little bit more complex a life cycle in that it produces those structures at the bottom there on the soil surface, which are the structures that give rise to the spores that then go up into the air. They can be important in all sorts of crops, so I see them in vegetable crops, but I did some work on cherries a few years ago and strangely was finding it as a fruit rot in cherries, and it suggests to me that there were those ascospores blasting up into the air. They're sort of under the right conditions, they will release and move up into the air, and then if they land on a flower, they can cause an infection, and you can see there the resulting fruit, or in the case of the tomato stem in the middle there, cause a rot. But they're also able to directly infect through the root system, and usually around the collar region of a plant. And that one on the left-hand side at the top is a lettuce plant, which is badly affected by Sclerotinia. But like I said, wide host range. There's a couple of different species. This one is Sclerotinia sclerotiarum, but there's another one as well that is again got a very wide host range. We even find it in Australia on native pastures and plants, so it's very widespread. Next slide, please. Just some interventions for Sclerotinia, and this is a very frustrating piece of research I was doing a couple of years ago, and one that I'm hoping we might get to in the baby leaf project, where we're looking at baby leaf lettuce and the again quite intensive production cycles. So we used in that case green manure, and we used oats, but I've also seen barley used as a green manure to, reduce the population. And so one of the effects of that, of those particular green manure crops, is to stimulate the microbial diversity in the soil. And there are some products, and I know in New Zealand you have trichodermas available. There's another one overseas called Aotearoa, which is a biocontrol agent. And what I was hoping, we got a 20% reduction in Sclerotinia in the treatments where we had the green manure with oats or barley, or it actually was oats in that particular trial, versus things like we used ryegrass as some, we had a bearfallow, a couple of others. There were some other green manure crops that we trialled, and we grew the lettuce on those after those were turned in, and the reduction was quite significant, but you could still see disease there. And I was really hoping that we were going to put some at the end of the crop, while the plants were still on the surface, and you could see the sclerotia forming on some of the plants that were infected. We were hoping to put a Aotearoa treatment across them, but unfortunately the farm was sold, so we never actually got that far. The other important thing, but I really think that would be the timing for those trichodermas, which would be able to then colonise those sclerotia sitting on the surface. And then fungicide application is the other important thing. So in a lot of crops, usually where they're transplanted, it's around transplanting time that the fungicide applications need to be made. Next slide, please. Look, I know the next presentation in this series will be on nematodes, so I won't go too much into them, other than in my experience, I guess I've come across 3 key kind of nematode diseases: root knot, cyst nematodes, and root lesion nematodes. That last one, root lesion nematode, I did some work years ago on potatoes where root lesion nematodes act in combination with verticillium, and I haven't really raised the issue of verticillium very much in this talk, but it's another important soil-borne pathogen. I see it in things like olive trees, almonds, we see it a bit. So it gets, again, across a wide range of plant species. But in potatoes, there's this strong interaction with root lesion nematodes, and it causes what we call early dying disease. So the crop starts to wilt and die prematurely, and so you just don't get tuber size. And it was one that at the time, it was over 20 years ago now, we were looking at fumigants to control it because it's very difficult to control. Verticillium also produces sclerotia, but they're not like the ones of Sclerotinia or sclerosum. They're tiny, they're microscopic, but they as well can survive for a long period of time. Another interesting interaction with verticillium and nutrition is that it's virtually the opposite to the effect that you get with the form of nitrogen in fusarium. So remember I said with fusarium, the ammonium form stimulates the fusarium disease. For verticillium, it's the opposite. Ammonium is suppressive to verticillium disease. So again, you need to know what your pathogen is so that you can manipulate your nutritional environment for them. Next slide. Next slide, please. Hello, Gina? Oh, thanks. We're getting towards the end now. So I did mention already disease complexes, and in some cases I was finding when I was looking at some of these lettuce crops, interactions between viruses, bacteria, some of those things like Pythium, various fungal and nematode pathogens. So it can be quite complicated when you're looking at soil-borne diseases and the pathogens that are causing them. I think it's a mistake sometimes to just look at them as single entities. Quite often they occur together, sometimes in succession, but more often I found that they were in complexes. So the diagnostic approach really does require to keep an open mind about what is there and what is the most significant, or what might be the succession of pathogens, and that might give some indication as to how best to manage the problem. But this particular case here, we were showing that tomato spotted wilt virus, which is obviously not a soil-borne disease, it's transmitted by thrips, was interacting with Pythium and causing, basically exacerbating those plants that had the virus started to die very quickly, but quite often they were associated with Pythium in that particular case. Next slide, please. So let's just start to sum up a little bit. Interventions, okay, so we always hear about biosecurity, sanitation and hygiene. So they're very important preventative things. And again, knowing what pathogens you might have on your property and what isn't there is really important. And there's been some incredible cases in Australia where I've seen the transmission or the movement of machinery and some of those viruses, those Tabamo viruses that I mentioned, with some of the machinery. And we know that, for example, things like clubroot, the clubroot pathogen can be transmitted generally, again, with either machinery or workers, or with flooding events, which we've had a few of recent times. Using suppressive organic amendments, cover crops and biofumigants, this is a huge area that probably needs more work. I'm still sort of trialling various things in these areas, and it's not an easy thing to get. It's no silver bullet, but it certainly can be helpful. There are some agrochemicals. Strangely enough, I was talking to one of the companies that have a product that will reduce both nematodes and fusarium as a soil drench, strange as it may seem. So I'm doing some trial work with that at the moment in melons. Improving soil physical and chemical health, really important drainage. In the work I'm doing in almonds with things like Phytophthora and some of the other diseases, drainage seems to be really crucial as a means of reducing the significance of pathogens that are probably very widespread in soils. pH, I've already mentioned a couple of cases where pH is important. For example, I said in clubroot. Microbial biocontrols and chemicals that induce systemic resistance, another huge area that I've done bits and pieces of work. It's very difficult to get funded work in those sorts of areas. I'm looking at some metal nanoparticles at the moment that act, well, supposedly act, there's some literature on it, that act to reduce some of these soil-borne pathogens and also induce some systemic resistance in plants to the attack. So that's another kind of an interesting area that's really probably only in the last decade as something that's come along, but lots of microbial controls available, and again, sometimes single microbes, or in some cases combinations of microbes. There's other various chemicals like silicates and humic acids and things that are all probably also biostimulants and forms of nutrient. So again, I mentioned those cases where the form of nitrogen can be important in terms of disease reduction, not control, but reduction. So it's part of an integrated package. Finally, the next slide. So let's just look at the main issues. There's many different types of pathogens. I've only mentioned a handful of them, but I think you'll be hearing in following webinars some of the others. Some are specific to certain family of plants. I mentioned some of those former specialises of Fusarium, the Sclerotium cepivorum in the Allium family. Some have a wide host range. I mentioned quite a few of those, the Pythiums, the Verticillium that have a wider host range. Some can be involved in disease complexes, so I've mentioned those. Some are favoured or suppressed by opposing types of nutrients, so again, the Verticillium versus Fusarium. Soil types and environmental conditions, so some are favoured in wet conditions, some are more favoured in dry conditions. I didn't really mention it, but there's some like Macrophyllina, another type of fungus that we've been finding associated with melon problems. It's more favoured in drier, hot conditions. So again, in some of our more arid areas, we see that as being an important soil-borne pathogen. Many can multiply by several orders of magnitude in a short period of time, and I referred to that when I talked about Pythium and Phytophthora. And most have a negative binomial distribution pattern. That means it's sort of like a bell-shaped curve. So sometimes over even a space of 10 or 20 centimetres, you can have a huge population in the middle, and out 20 or 10 centimetres away, a very low population. And that makes that problematic when you're looking at trying to measure the population in the soil and determine whether or not it's significant or not. You really need to take a lot of subsamples. You need to be aware of organic matter that might be housing billions of bits of DNA anyway, if you're using DNA technology to measure these things. So I might finish it there. I think I've just got the final slide to take questions. Thanks.
That's fantastic. Thanks, Len. That's a wealth of information, and yeah, and it's great to see what you've done through your career and your understanding of the topic. We have got a few questions because it's very much a challenging topic for us here in New Zealand as well. So I might just read out a couple of questions for you. From Melania, would an ammonia-based fertiliser also impact Fusarium in a root rot complex in the same way as increasing wilt symptoms?
Yes,
ammonia-based
ammonium.
fertiliser.
I think that was the point I was making, that the ammonium fertilisers tend to favour the Fusarium. It does a few things. It sort of reduces the pH slightly, but it also has a direct effect on, and again, it's in the literature, but it's kind of hard to interpret the exact mechanism, that it actually will make the Fusarium more aggressive and the plant more susceptible. So it's sort of a combination of favouring the pathogen and making the plant more susceptible to the symptom development anyway.
Yeah, and it's something you've seen in a root rot complex as well, or is it just the straight Fusariums that tend to express it?
I think most of the time I'm looking at root rot complexes anyway. So if I do a diagnosis of a say a melon that's wilting, I'll find some Pythium in the finer roots, and I'll find some Fusarium in the vascular tissue. So quite often and we'll find Macrophyllina or sometimes even the gummy stem blight fungus. So there's quite often they are in complexes. I guess what I don't normally see is a Fusarium wilt pathogen and a Verticillium together. Generally, they are in separate root disease complexes, and yeah, so that's pretty much it.
Cool, thank you. Next question, can you discuss factors that allow Fusarium to become virulent within the plant versus not, for example, in tomatoes? So once again, Fusarium crops up.
Well, I guess, again, nutrition is going to be important. It's quite a universal thing for the Fusarium oxysporums that you'll find a lot of literature where there's that interaction with the form of nitrogen. But I guess using resistant rootstocks is what we've used most successfully for things like tomatoes. So there are resistant rootstocks available, which both have resistance to the Fusarium, but also impart strong vigor in the scion, so that you can actually quite often crop two heads on the one plant root system.
Great, thank you. Next question is, what advances have you seen in using diagnostics to help with in-field management? Has it advanced, or are we still at the point where we're going, yeah, we can diagnose it, but we can't really use that to help us that much?
I think it's a tricky one, because I don't want to bag the people that are looking at trying to predict. I think it's a useful tool. Certainly from a research point of view, it's a useful tool when you're looking at interventions and you want to compare an intervention versus no intervention, or one intervention versus another. But like I said, there's a big dilemma in my mind in terms of how these soil-borne pathogens are distributed. And so it probably requires a sort of a sampling methodology that's specific to the particular pathogens you're looking for, and also knowledge of other factors within a field, where that virtually every field there'll be some areas where it doesn't drain as well, or there's a shallower lens of soil in some areas or whatever. So it's sort of the whole uniformity thing, I think, is really a critical factor in understanding or using those things as tools. Diagnostics is important in the longer term, though. If you've got, for example, a new pathogen that's turned up, it's really important to know as soon as possible. And unfortunately, some of the things I've found in my career, they've been around for a year or two before the diagnosis was made. And that's where, quite often the whole issue with biosecurity comes unstuck, because, we have government processes that say, oh, we've got to do this and contain it and all this sort of thing. It upsets growers because they get quarantined. It just leads to a whole problem, and quite often it's the horse is bolted by the time it's been detected, and it takes a while for us to catch up.
Yep.
So, yeah, I think monitoring and surveillance would be where I'd be putting my money. Yeah.
Sure. Okay, and again, that's another topic, and later in the series where we do a bit more of a deep dive into diagnostic techniques and so forth, and where to use them and how to use them. So that's great. Thanks for your overview of that, Len. Just one more question here, quite specific, Fusarium and squash. What recommendations do you have around biostimulants and biologicals to help with that particular soil-borne disease issue?
Tricky one. I haven't done any research on the squash myself, so it's a bit tricky. And again, it's unlikely that it's a Fusarium oxysporum wilt pathogen. It may be more like a cholera type pathogen. So again, I haven't not worked on it. It's a bit tricky to say. Generally, though, and I mean, I'm not saying it's not a true fusarium wilt. We use squash in Australia as a rootstock to control things like fusarium. So it certainly, they have their benefits. But the whole issue with biostimulants and the like, I think it needs research. There's no silver bullet. As I said, it's something that they may be. It's working out a good use pattern like any product, even agrochemicals. You've got to understand the use pattern for them to get the best out of them. So really, it does require research to really understand how best to use them and when to use them.
Well, thank you. That's the questions we have online there. So just wrap up now. Thank you very much, Len, for the talk today and sharing your knowledge with us. You've got a wealth of knowledge there, so really appreciate you coming online, joining us from New South Wales today. And just remember, the next webinar in the series is next Wednesday, the 17th of September, again at 10 a.m. So we'll look forward to you all joining us again for that. And yeah, just say once again, thank you very much, Len, and yeah, look forward to seeing you all next week.
Thanks, everybody.